This presentation is about proteins and enzyme function. We'll begin with proteins. You should know this. Proteins are made of what monomer? I hope you said amino acids. Where do all these amino acids come from that organisms use to build their proteins? Let's start with the plants. Plants, of course, don't eat other food. They are autotrophs. So they must make their own amino acids. They do this with the simple sugars that they make through photosynthesis, add them to nitrogen, and use these components to build their own amino acids. Humans can make some amino acids, but not all of them. And the others we must get from our food. Protein is somewhat unusual compared with carbohydrates and fats in that we don't store it. So we do need to eat proteins each day in order to get the amino acids we need to build proteins. It's important to point out that proteins are in every food that we eat, in different quantities and types. That means that if you are a vegetarian or even a vegan, that you can get enough protein by eating the right kinds of plant foods, particularly foods like legumes, soybeans, beans, whole grains, things like that contain a high level of protein. Amino acids have a very specific structure. Notice that on one end there is an amino group with the nitrogen, and on the other end a carboxyl group. These two parts of the amino acid are the same on every single amino acid in a protein. However, each type of amino acid has a different R group, a different little group of atoms attached in the middle. The reason this is important is because when the amino acids are connected to each other, they fit together front to back. And hundreds or even thousands of amino acids can be connected in this way. Once all the amino acids are connected together, there is a sequence of different R groups, which have different properties. And the sequence of these different R groups on the amino acids cause the whole chain of amino acids to fold in a particular shape. This is an image of different amino acids. You don't need to know them all. Just notice that each amino acid has an amino end and a carboxyl end that attaches to the next amino acid. You can see the variety of different R groups that are attached to this basic front and back end. When amino acids are put together to form a protein, there are specific reactions that occur. When putting two amino acids together to form what we call a dipeptide, simply means two amino acids, two hydrogen atoms and an oxygen atom are removed, and a bond called a peptide bond is formed. Peptide is another word for amino acid, and it is a term that is used when proteins are being put together or taken apart, as in digestion. This type of chemical reaction is called condensation, because water is formed, or dehydration synthesis, meaning that the water is removed and a larger molecule is synthesized or built. This process continues over and over to build a longer and longer chain of amino acids. This chain is called a polypeptide, and a polypeptide can be hundreds or thousands of amino acids long. Once the chain of amino acids has formed, it begins to fold into predictable patterns. That is the secondary structure. Eventually, this forms a very complex three-dimensional shape, and in some cases combines with another protein to form an even more complex molecule. This process happens in your cells. It is called protein synthesis. And there are structures called ribosomes in your cells, sort of like super enzymes, that connect each amino acid to the next. Again, it is important that the amino acids be in a very particular order, what we call a sequence, because that sequence determines how the protein will fold and what function it will carry out. What makes it fold in a particular way are those R groups pointed out before. For example, if an R group is hydrophobic, it will tend to fold to the inside of a protein to be away from the water in the cell, whereas hydrophilic R groups will tend to fold to the outside of a protein to be near the water in the cell. Here is a big idea in biology. You have heard that genes help to cause your traits. The way that it works is that your DNA has basically instructions for putting together amino acids in a particular sequence to form particular shapes of proteins. And it is the shape of the protein that determines its function, what it is able to do. So think about it. If you change the DNA, we call that a mutation. What do you think that would happen, or what do you think that would cause to the shape of a protein? You should have thought something like the following. If the DNA or the sequence of the DNA itself is changed, a mutation, then that provides a different set of instructions. In turn, ribosomes will put together a different sequence of amino acids, and that chain of amino acids may fold into a different shape. If so, the protein may not have the same function. In many cases, the protein will not work at all. However, in rare cases, the protein may actually be able to carry out a different function. This provides the raw material for evolution. So why think so much about proteins? Proteins are important to us for both structure and function. Remember that structure is the matter that we're made of, and much of the matter that we're made of is protein. For example, think about your muscles. And you may not realize it, but other components of your body, skin, bones, etc., also contain a great deal of protein. In addition, proteins are actually able to do things, to move, to make other molecules do things, to send messages to other molecules and other cells. Because proteins are so important to biology, you will need to know many protein functions of common proteins. Practice them frequently so you feel comfortable using the names in context and describing what they do. Here's another way to look at it. Proteins can cause different traits in three basic ways. They can make up our physical structure, and if those structures are different from person to person, those might be different traits. Proteins can cause chemical change. That means that enzymes, which is a type of protein, are causing chemical reactions to occur. And third, proteins can cause non-chemical change. This basically means anything that is not an enzyme causing a chemical reaction and not structure. So for example, proteins may be able to send messages to other cells or other organs. Proteins may be able to transport other molecules around a cell or through a cell membrane. Here is a list of common proteins and their functions. Don't worry so much about the category. Focus on the names of the proteins and what they do. So for example, collagen is the most common protein in the human body. It makes up a good part of many of our structures, as you can see on the list below. Now, structures, of course, can be made up of many things. For example, skin contains collagen, elastin, and keratin. Bones contain collagen, calcium, and blood vessels that run through the bone tissue. Some proteins are combined with other kinds of molecules to do their work. For example, hemoglobin is protein with an iron atom on it, and this allows hemoglobin to very effectively carry oxygen inside of our red blood cells. Antibodies are a type of protein made by our immune system, and they are able to recognize the antigens or markers on the outside of cells. In this way, they are able to identify pathogens, microbes such as bacteria and viruses, and immobilize them. Enzymes catalyze chemical reactions. We'll talk more about those in a bit. Some hormones are made of protein, not all. Other hormones are steroid types of hormones, and these molecules are able to send messages to other cells and tissue. In turn, receptors on cells receive those messages by having shapes that match the hormones or are complementary to the shape of the hormones. Other types of functions include cell transport, moving molecules through cell membranes or moving molecules around cells. Very specialized molecules such as actin and myosin that are able to physically move with energy from ATP, and this is how our muscles move, as well as how molecules are moved around within cells. We will also talk later in the class about histones, proteins within our chromosomes that help to regulate the actions of our genes. Now, a bit more about enzymes in particular. Enzymes are what we call biological catalysts. That means that they are molecules within living things that cause chemical reactions to occur. Now remember, in a chemical reaction, there are molecules called reactants, and they are combined and converted or broken down or built up and changed into products by forming and breaking chemical bonds. Chemical reactions do not have to occur in living things. There are many other chemical reactions that occur in the non-living world. If the chemical reactions occur in living things, they are generally catalyzed or triggered by an enzyme. And in that case, we call the reactant a substrate. So an enzyme acts on a particular substrate. So we say that enzymes help to convert substrates to products. This is very important for living things because living things need to be able to control the chemical reactions that occur. They need to be able to make the chemical reactions happen quickly, and at particular times, and then to stop happening when they are no longer needed. Living things could not function without their enzymes. Remember that all these chemical reactions, building up molecules and breaking down molecules, are all part of our metabolism. So a general way to think about enzyme function is in two categories. Some enzymes put together substrates to form a product. An example of this is when a plant combines glucose molecules into long chains of starch. We say that it is building monomers into a polymer. A continuous chain of glucose is built into a polymer of starch. In other cases, enzymes break up a substrate into smaller molecules. So for example, organisms that contain the enzyme amylase can break down a polymer such as starch into its individual monomers of glucose. This happens when we digest starch. Here's a helpful hint. You can often recognize enzymes, even if they have very long names, with their suffix ase. In this class, we will refer to two common enzymes in many different examples, lactase and amylase. Lactase breaks down lactose. Amylase breaks down starch. The chemical name for starch is amylose. That's why this enzyme's name is amylase. Now remember that enzymes are proteins, and proteins fold into a particular shape to have their function. An enzyme is a protein that has a particular shape on one part called the active site. This is the specific area on the enzyme where the substrate will bind. Bind means a sort of light attachment. The substrate binds to the enzyme in part because it has a complementary shape. Complementary means not exactly the same, but sort of like puzzle pieces. They fit together. Enzymes are able to catalyze chemical reactions in very physical ways. They literally can hold the substrates next to each other for a brief period of time, allowing those substrates to be more likely to form a reaction with each other. Or perhaps they stress the molecule in such a way that it is more likely to break apart. Many reactions, generally the ones where molecules are built or synthesized, require ATP to occur. In this case, enzymes work with a nearby molecule of ATP to provide the energy needed for the reaction to take place. You can think of enzymes as tools. Imagine a stapler. I can staple paper over and over and over again. It doesn't change the stapler in any significant way. Perhaps if I used it a million times, I would need a new stapler. But in general, I can staple, 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 and use that same tool over and over again. Similarly, an enzyme can catalyze multiple reactions with multiple substrates over and over before it is broken down and discarded by the cell. Many enzymes may sort of change what we call conformation or shape during the reaction, but then they return to their original form, similar to the way that a stapler changes in position and then returns to its original position to staple again. Many enzymes are specialists, meaning that they only work with one substrate and only catalyze one reaction. In some cases, enzymes are what we call generalists, meaning that they can catalyze multiple reactions, usually similar types of reactions. You can think of this as your basic stapler can staple different types of paper, but perhaps I would need a specialized stapler to staple thick cardboard. There are many factors that affect how enzymes function. These factors can relate to concentration or the environmental conditions around the enzyme. There are 4 factors that you will need to know. The first one is enzyme concentration. The idea here is that if there is a certain amount of substrate, but more enzyme is added, the rate of reaction, how quickly the substrate is converted to product, will increase. Let's use an example. Say that you've eaten a big plate of pasta, and you chew up the food and you swallow it, and it goes through your stomach and it goes to your intestines, and you have many molecules of starch in your intestines. Your intestinal cells will begin to produce the protein amylase through protein synthesis. The amylase will start to react with the molecules of starch. Starch molecules will randomly collide with amylase, and the amylase will break the bonds holding the glucose monomers together. In response to the presence of starch, intestinal cells will produce more and more enzyme. As the concentration of amylase increases, the rate at which the starch is broken down can increase as well. This may go on until the substrate has all reacted, in which case the cells would stop producing amylase. However, in most cases, the cells do not continue to produce the enzyme indefinitely. And at some point, the cells produce just a limited amount of amylase in this case. There is often much more substrate, so perhaps many more starch molecules than amylase enzymes. In this case, as the concentration of starch molecules increases, there comes a point at which every amylase molecule is colliding with and catalyzing reactions and binding to the starch molecules all at once as quickly as they can, meaning each molecule of amylase is bound to a molecule of starch or in the process of colliding and binding with that molecule at any given time. So the rate of reaction cannot increase. You can see in the graph that the line levels off. That means that the rate of reaction increases at first as more starch is added, but then once the amylase molecules are physically filled up and occupied, we say that they are saturated and can no longer catalyze the reactions any faster, and the reactions cannot occur at a faster rate. Another way to think about this is that the enzymes are the limiting factor to this particular reaction. The amount of substrate, or the concentration of the enzyme, in this case amylase, is the limiting factor to how quickly the starch can be digested. Something you need to know about enzymes is that they must have certain conditions in the environment in order to function. If the conditions become too extreme, the enzymes may denature. What this means is that chemical bonds within the enzyme start breaking down, and the shape of the protein changes. This changes the shape of the active site of the enzyme, and it no longer fits with the substrate and therefore cannot bind. This is why cells, or at least one reason, why cells must maintain homeostasis. They must maintain quite narrow ranges of conditions such as pH and temperature. If not, their enzymes would denature and they would no longer be able to function. You've actually seen the effects of denaturing when you cook food. Think about cooking an egg. It starts out as a liquid, and as you heat it up, it suddenly changes into something more solid with a different color and texture. That is because the proteins in the egg have denatured as a result of the high cooking temperature. pH also has an effect on enzyme function. If enzymes are exposed to extreme levels of pH, too acidic, too basic, the enzymes may denature. Now, most enzymes function well at the pHs most commonly present in living things, near a neutral pH. There are, however, cases in which enzymes function at very different pHs. They may be specialized to work in quite extreme conditions, for example, inside of your stomach or inside the lysosomes of your cells. This is very important as it allows your cells in your body to isolate certain very powerful enzymes because they only work in a particular set of conditions in a particular location. As mentioned with the example of cooking, temperature affects enzyme function as well. Notice on this graph that the slope on either side of the curve is quite different. On the left, the slope is more gradual. This is because increasing the temperature gradually increases the motion of the molecules so that the substrates collide with the enzymes at a faster rate, increasing the rate of reaction. However, on the right side of the graph, the slope is more steep. This indicates that the enzymes stop functioning fairly suddenly. This is because when the conditions reach a certain temperature, the enzymes rapidly begin to denature and can no longer function at all. Again, this is why cells must maintain homeostasis to keep pH and temperature within the optimal range. So, what do you need to know about proteins and enzymes? Make sure you can describe the general structure of proteins, amino acids, and how they fold. Common proteins, the names and functions shown in this presentation. Enzyme structure and function, particularly described by the terms active site, substrate, and product, and the factors affecting enzymes, including enzyme concentration, substrate concentration, pH, and temperature. Make sure that you can describe each of the enzyme graphs and explain what it means for enzymes to denature. Make sure that you ask any questions that you have about this material, and practice. Bye-bye.